Hi, Baxter. Today I'm going to walk you through how to extract DNA from your own body. You should have all received your kit by now, and also some instructions with some important thought questions related to this experiment.
All right, so to collect your cells, you're going to use this pipette, and you need 3 milliliters of water. This is 1 milliliter. 2 milliliters. 3 milliliters.
Set the water aside and chew on the side of your cheek for 30 seconds. Now grab your water, rinse, and spit.
All right, so I've got my cheek cells in here, and I'm going to take this, which you all have. It's a lysis buffer solution, and I'm going to pour the liquid, which contains my cheek cells, into the solution. All right, so now you're taking this. This is your protease. It's in this little tube. It's a powder, and you're adding 0.25 milliliters of water to this solution. So I have it measured out. It's in the pipette. All right.
Did you know that meat tenderizer you buy in the store is a protease? It breaks down the protein in your steak. After mixing the protease solution, add it to your cheek cells solution. Invert several times to stir. If you were at school, you'd be using this as a water bath. It just can kind of set the temperature and keep it there. So we want the temperature at 50 degrees. You have a thermometer at home. That was in your first care package. You can do this in your sink. And you're gonna now let your solution of cheek cells in the buffer soak in this bath. It also has the protease solution in there. And that's going to basically free the DNA from the cells. So that's happening for 10 minutes. I created that styrofoam float thing so that it'll just float in there. After 10 minutes in the warm water bath, you're going to carefully add the alcohol.
All right, so I took this alcohol out of the freezer. You want it to be icy cold, and we're going to get this alcohol into this solution, but you don't want to pour it in. You want to have it in at about a 45-degree angle, and the alcohol is going to sit on top of the solution. So, oh yeah, it's working. It's like sitting on top of it. So I'm going to do this with most of the alcohol.
So you can see the lysis buffer down the bottom and the alcohol layer up at the top. The DNA is going to precipitate into little clumps where this interface is. So you'll stick your pipette down into that place where the two layers meet and extract the DNA.
All right, so 5 minutes have gone by.
So I can see small little white strands floating right at the place where the alcohol and the buffer meet. And this is a better picture for you to see than mine. So I wanted you to see that's a strand. That's a DNA strand. So you're going to use your pipette to reach down inside the tube and extract a few of these strands for your necklace.
And now I have a DNA necklace. I don't know if you can see it. Have fun.